Hello, dear students. In this video, we are going to learn about sex linkage. At the end of this lesson, you will be able to explain how sex chromosomes determine biological sex, compare sex-linked and sex-influenced traits, solve genetic problems, and describe the role of recombination in inheritance. This is this lesson's essential question. What is the role of sex linkage, and how does recombination affect genetic variation? Sexual differentiation is the process by which physical differences between males and females develop. It occurs after gonadal determination, when hormones begin to act. These hormones influence the development of the external genitalia, internal reproductive organs, breasts, body hair, and other secondary sex characteristics. The XY sex determination system is used by humans and most mammals. In this system, biological sex is determined by the sex chromosomes. Females typically have 2 X chromosomes, or XX, while males have 1 X and 1 Y chromosome, or XY. The Y chromosome carries genes that trigger male development, which is why the father determines the sex of the child. Sex determination does not occur the same way in all organisms. Some insects use the XXXO system, where males have only 1 X chromosome. Birds and some reptiles use the ZWZZ system, where females are ZW and males are ZZ. In certain reptiles, such as turtles and alligators, the temperature of the egg during incubation determines whether the offspring becomes male or female. Karyotyping involves photographing chromosomes during metaphase, when they are condensed and easy to see. At first, the chromosomes appear scattered. Scientists then arrange them into homologous pairs to form a karyotype. This method is used to identify chromosomal abnormalities, such as extra chromosomes or missing chromosomes, associated with genetic disorders like Down syndrome. A karyotype is an organized picture of all the chromosomes found in a cell, arranged by size and shape. Humans normally have 46 chromosomes or 23 pairs. Pairs 1 to 22 are autosomes and are the same in males and females. The last pair are the sex chromosomes. Males usually have XY, while females have XX. Karyotypes are useful for studying chromosome structure and identifying abnormalities. Let us look at how sex develops over time. Sexual development happens in 3 major stages. First is chromosomal sex, which is established at fertilization when the egg and sperm combine. Second is gonadal sex, when the embryo's gonads develop into either testes or ovaries. Third is phenotypic sex, which refers to the physical traits that develop under the influence of hormones, including internal reproductive organs and external genitalia. This shows us that sex development is a process, not a single event. In early development, all embryos start the same way. They have undifferentiated gonads and both sets of internal reproductive ducts, the Wolffian ducts, which are associated with male development, and the Müllerian ducts, which are associated with female development. Around 8 to 9 weeks of development, gene expression begins to direct development. If the SRY gene on the Y chromosome is present, the gonads develop into testes. If the SRY gene is absent, the gonads develop into ovaries. This explains why early embryos do not show clear male or female characteristics. Now let us look at what happens when this process is disrupted. Some mutations affect how the body responds to hormones. One example is complete androgen insensitivity syndrome. In this condition, an individual has XY chromosomes and forms testes, but the body cannot respond to male hormones. Because the hormones cannot act, external development follows the female pathway. This shows us that hormones and receptors are just as important as chromosomes in determining physical sex traits. This diagram explains how mutations can alter phenotypic sex, even in individuals with XY chromosomes. These conditions are collectively called XY disorders of sexual development, XYDSD. Although XY chromosomes usually lead to male development, this diagram shows that chromosomes alone are not sufficient. Normal sex development also requires functional genes, hormones, and hormone receptors. In the top row, labeled simulating males, individuals have XY chromosomes and develop testes, but development is atypical. The testes may be located in the abdomen or inguinal region rather than fully descended. Because hormone production or hormone action is partially impaired, male development occurs, but may be incomplete or abnormal, showing that the presence of testes alone does not ensure typical male anatomy. In the bottom row, labeled simulating females, individuals also have XY chromosomes and testes, but androgen hormones cannot act effectively. As a result, external development follows a female typical or ambiguous pathway. This highlights a key concept. Phenotypic sex depends on hormone response, not just the presence of a Y chromosome. These conditions are caused by mutations in different genes involved in sex development. Terms such as pseudohermaphroditism were used in the past, but today disorders of sexual development or gonadal intersexuality are preferred. Early in life, phenotypic sex may not match chromosomal sex, and physical characteristics may change as development and hormone activity progress. This table shows us that biological sex is determined by several factors, not just one. These include chromosomes, gonads, genitalia, and other physical characteristics. Most people fall into the typical patterns of XX females and XY males, but the table also shows natural variations. Some individuals have chromosome combinations such as XXX, XXY, XYY, or only one X chromosome. These variations can affect fertility, physical traits, and development. The key idea here is that sex is biologically complex. Chromosomes, organs, and hormones do not always align in the same way for every individual. Some traits occur more frequently in one sex than the other because they are sex-linked. This means the genes controlling these traits are located on the X or Y chromosome. Conditions such as red-green color blindness and hemophilia are more common in males because males have only one X chromosome. The X and Y chromosomes are known as sex chromosomes because they play a key role in sex determination. Sex linkage refers to the expression of traits controlled by genes on these chromosomes. X-linked traits are more common and affect both sexes differently, while Y-linked traits appear only in males and are passed from father to son. This table summarizes several human genetic conditions that are X-linked, meaning the genes responsible for these conditions are found on the X chromosome. The table groups these conditions into X-linked recessive and X-linked dominant inheritance patterns, and it also shows the genetic cause and the main characteristics of each condition. Let us start with the X-linked recessive conditions listed at the top of the table. In these conditions, males will express the trait if their single X chromosome carries the mutation. Females usually need 2 copies of the mutated allele to be affected, so they are often carriers. Now let us look at the X-linked dominant conditions listed in the table. In these cases, only one mutated allele is needed to express the condition. This means both males and females can be affected, although the severity may differ. X-linked traits are caused by genes found on the X chromosome. Males have only one X chromosome, so a single recessive allele is enough for the trait to be expressed. Females have 2 X chromosomes, so they often carry the allele without showing the condition. This explains why conditions like hemophilia and color blindness occur more frequently in males than in females. These images show the Ishihara color blindness test, used to detect red-green color vision deficiency. Each circle is made of colored dots that form numbers. People with normal color vision can see the numbers clearly, while those with red-green color blindness may see different numbers, or none at all. This condition affects how certain colors are distinguished. These Punnett squares show the inheritance of X-linked recessive traits, using red-green color blindness as an example. Fathers pass their X chromosome only to daughters, while mothers pass an X to both sons and daughters. Sons express the trait if they inherit the recessive allele, while daughters usually become carriers unless they inherit 2 recessive alleles. This infographic illustrates the inheritance of X-linked disorders, using Duchenne muscular dystrophy as an example. Because males inherit only one X chromosome, a single mutated allele can cause the condition. Females have 2 X chromosomes, so they are often carriers and may not show symptoms, but they can transmit the allele to offspring. Let us work through this X-linked inheritance problem step by step. In this example, we are studying color blindness, which is an X-linked recessive trait. We use X with capital letter B to represent the allele for the dominant normal vision, and X with small letter b to represent the allele for the recessive color blindness. Next, we identify the genotypes of the parents given in the problem. In the first example, the female is heterozygous XBXB. The male is normal, so his genotype is dominant XBY. In the second example, the female is normal and non-carrier, so her genotype is dominant XBXB, while the male is colorblind with recessive genotype XBY. We then fill in the Punnett square by combining the gametes from the mother and the father. Each box represents a possible offspring genotype. After filling in the Punnett square, we identify each offspring's genotype and determine the phenotype. Some offspring will be normal females, some will be carrier females, some will be normal males, and some may be colorblind males. Remember, females must have 2 recessive alleles to be affected, while males need only 1. Finally, we count the outcomes. Since each combination appears once, the genotypic ratio is 1, is to 1, is to 1, is to 1. Even though the genotypes are evenly distributed, the phenotypic ratio is 2, 1 to 1. Let us work in this next problem. We are still studying color blindness, which is an X-linked recessive trait. Next, let us identify the parents and their genotypes. The female parent is normal and non-carrier, so her genotype is dominant XBXB. The male parent is colorblind, so his genotype is recessive XBY. When we fill in the Punnett square, we combine these gametes. All daughters receive dominant XB from the mother and recessive XB from the father, giving the heterozygous genotype XBXB, which means they are carriers. All sons receive dominant XB from the mother and Y from the father, giving the genotype dominant XBY, which means they are normal males. Finally, we count the outcomes. Since each combination appears twice, the genotypic ratio is 2:2, simplified to 1:1. The phenotypic ratio is 2:2, simplified to 1:1. Y-linked inheritance, or halandric inheritance, involves traits carried on the Y chromosome. Since females do not have a Y chromosome, these traits appear only in males. An important pattern is that an affected father passes the trait to all of his sons, but to none of his daughters. Y-linked traits are rare, because the Y chromosome contains relatively few genes. Here are examples of traits linked to the Y chromosome. Hypertrichosis involves excessive hair growth in certain areas of the body. 47XYY syndrome, also called Jacobs syndrome, results from an extra Y chromosome and may be associated with increased stature and learning challenges. Some inherited eye conditions, such as certain forms of retinitis pigmentosa, have also been linked to Y chromosome genes. Sex-influenced inheritance refers to traits controlled by autosomal genes, but whose expression depends on sex hormones, such as testosterone and estrogen. These traits can be seen in both sexes, but they occur more frequently or more strongly in one sex. Pattern baldness is a well-known example, appearing more commonly and more severely in males. This slide highlights several conditions influenced by sex. Clubfoot occurs about twice as often in males. Gout, caused by uric acid buildup, is much more common in males. On the other hand, osteoporosis, which involves bone weakening, and systemic lupus erythematosus, an autoimmune disease, are more common in females. These differences are largely influenced by hormonal and physiological factors. Sex-limited inheritance involves traits that are genetically present in both sexes, but are expressed in only one sex. This often occurs because of differences in reproductive organs or hormone levels. The examples shown illustrate sexual dimorphism, where males and females of the same species display clear physical differences. These examples show sex-limited traits and sexual dimorphism in different species. In the spider, the female is much larger than the male, which is common in many spider species. In map turtles, males and females differ clearly in body size. The fish example shows a tiny male attached to a much larger female, and in birds, males are brightly colored while females are duller. These traits are expressed in only one sex, or differ greatly between sexes. Genetic recombination occurs during meiosis, starting with crossing over. This process happens in prophase I when homologous chromosomes exchange segments of DNA. Crossing over creates new combinations of alleles, increasing genetic variation in the resulting gametes. Another source of genetic recombination is independent assortment. During metaphase I of meiosis, homologous chromosome pairs line up randomly at the equator. This random arrangement produces different combinations of maternal and paternal chromosomes in gametes. The third source of genetic recombination is random fertilization. Any sperm can fertilize any egg, and each gamete is genetically unique. The combination of two unique gametes creates enormous genetic variation among offspring. Genetic recombination is important because it increases genetic diversity within a population. By reshuffling genes, organisms produce unique combinations of traits. This variation helps populations adapt to changing environments, survive diseases, and drive the process of evolution. To answer this lesson's essential question, sex linkage affects inheritance by determining the transmission of traits linked to sex chromosomes, while recombination increases genetic variation by exchanging genetic material between homologous chromosomes during meiosis. To conclude, name the chromosomes that determine biological sex in humans, describe one example of a sex-linked trait, and explain how recombination contributes to genetic variation. Thank you for listening.